Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. So we already discussed the MHC 1 molecule and MHC 2 molecule, means the big picture, the overview of MHC 1 and MHC 2 molecule, and we also discussed the structure of MHC 1 and MHC 2 molecule. Now this is another video lecture which is also regarded to the MHC 1 and MHC 2 molecule, and in this video lecture we are going to talk about the processing of MHC 1 molecule, and inshallah in the next video lecture we are going to talk about processing of MHC 2 molecule, means the synthesis of MHC 1 molecule. So let me write synthesis of MHC 1 molecules, or we can say receptor. Okay, now in the before going to explain this synthesis or processing of MHC 1 molecule, you guys need to understand about the structure and the overview of MHC 1 molecule. Once you get the idea about that, what are MHC molecules and what are the structure of MHC molecule, so then this one video will be easy for you. Okay, so it is present into my channel, you can watch it, or don't worry, I will give the link in the description box of the previous videos, then you can watch it. Now let's start. Synthesis of MHC1. Now, how the MHC1 molecule are synthesized, how it is processed. This MHC1 molecule, as I already told you, that it is usually present in all nucleated cells. Okay, so let's say we have a nucleated cell. In this case, let's take it, it may be the tissue cells or any cell of the body which are nucleated. Okay, means how this MHC1 molecule are synthesized. For the synthesis of MHC1 molecule, we need 3 or almost 4 structure of the cell. We need cell membrane structure, we need the endoplasmic reticulum, we need the Golgi body, and we need proteasomes, and some other molecules are also. So we need these kinds of structure of the cell through which the cell can easily synthesize this MHC1 molecule. So let me draw each of the structure of the cell which I told. So let me draw. First of all, let me draw the cell membrane here. So let's say this is the cell membrane of eukaryotic cell. So let me write this part. This is the eukaryotic cell, or we can say, let's say this is the nucleated cell. Eukaryotic cell. Okay, now outside the cell membrane we have extracellular fluid, and inside the cell membrane we have intracellular fluid. Okay, now inside the cell membrane we have what? We have the so I not draw the complete structure of the cell membrane here. So just for your understanding, I draw a little bit or a small structure of the cell membrane. Now inside the cell we have Golgi body and endoplasmic reticulum. So let me draw the endoplasmic reticulum. So here I am not drawing the exact structure of the endoplasmic reticulum, just a rough structure of the endoplasmic reticulum, that how it will look like. So let's say, usually in this case we usually take the rough endoplasmic reticulum. Okay, so let me draw. Okay, let's say this is the endoplasmic reticulum, and this is what? This is the rough endoplasmic reticulum. Okay, rough endoplasmic reticulum. And all of you better know that rough endoplasmic reticulum contain ribosomes, a lot of ribosomes. So here I will draw just one ribosome. Now this is the large subunit, and this is the small subunit of the ribosomes. Okay, now after that we have another structure of the cell which are specially responsible in the synthesis of MHC1 molecule. We call that Golgi bodies. So let me draw. Now we have another structure which is also need for the MHC1 molecule. We call that proteasome. So let me draw. Proteasomes. Okay. So these are the structure which are specially need for the MHC1 molecule. Although the endoplasmic reticulum connected with the nucleus, but I not draw the nucleus. I reject the nucleus. Why? Because nucleus are not needed for the MHC1 molecule nucleus. Although this is the nucleated cell. Okay, now keep it in mind. So what actually happened after that? After that we have a pathogenic bacteria. In this case, let's take a pathogenic bacteria. Pathogenic bacteria. Now this bacteria is get internalized. It is present in extracellular fluid, and it get internalized and reach to the intracellular fluid by the cell membrane. So now it get internalized. So once this bacteria get internalized inside the cell membrane, so after that what actually happen? This bacteria will produce, they use the cell machine and produce their own protein, which we called it endogenous protein. So let's say they produce their now once they form endogenous protein, each proteins are then passes from the proteasomes. Now proteasomes will break down and convert each protein into 15 amino acid. Why? Because all of you better know that protein is actually made up of amino acid. So when the protein are break down, so it is converted into amino acid. So this is 15 amino acid. Okay, after that these 15 amino acid are passes through the endoplasmic reticulum, means rough endoplasmic reticulum, through a channel, through a way, through the passage, which we call it tape transporter. Let me draw here. Now once the protein are passes from the tape transporter, so then these proteins once these amino acid are passes from the tape, let me write, these are the tape transporter. So each amino acid are further converted into or each amino acid are further hydrolyzed, and we get 8 to 10 amino acid. Now after that, these ribosomes are activated and they make the product, or they make the protein, which are specially responsible for the MHC1 molecule. So okay, now once we get the protein of MHC1 molecule, or the product of MHC1 molecule, we get incomplete MHC1 molecule. Let me draw. As we discussed that MHC have 4 chain, 4 domain: alpha 1, alpha 2, alpha 3, and beta domain. Okay, so first let me draw the, okay, this is the alpha 3 domain. Now after some time we get this incomplete structure of the MHC1 along with a protein which we call that calexin. Now this is another protein which is also formed. We call it calnexin. Now this protein is just like, they are acting as a chaperone, and this protein will bring this MHC molecule inside the endoplasmic reticulum. Now we get the incomplete we get the incomplete MHC1 molecule. After that we also inside the endoplasmic reticulum there is a beta 2 subunit. This beta 2 subunit are joined with this alpha 3. Now once it is joined with alpha 3, we get a complete MHC1 molecule, or we can say a mature MHC1 molecule. So we get a complete MHC1 molecule. Now let me draw a complete MHC1 molecule here. Beta. Okay. Now this is the complete MHC1 molecule. After some time, there is another protein which bind to this binding side of antigen or to this groove. So we call that, this is another protein. We call that ERP57. So we get ERP57 that bind to this, that just attach to this binding side of the antigen or the groove of MHC1 molecule to prevent the antigen which are binding to that MHC1 molecule. Now once we get this ERP57 protein, so this is actually this beta 2 having weight, they have 12 kilo Dalton weight, this beta weight, and this one molecule as you are watching, they have this incomplete MHC1 molecule having weight 42 kilo Dalton, 43 kilo Dalton, and completely we get 55 kilo Dalton. So completely we get 55 kilo Dalton. So this is the 55 kilo Dalton MHC1 molecule. So we get a complete MHC1 molecule having ERP57 proteins to prevent the antigen to bind this groove. Now after some time, this complete MHC1 molecule are transported and shifted to another protein which we call the tepsin. Now what actually tepsin doing here? Let me write here. So let's say these are the tepsin molecule. These are the chaperone. And this is the complete MHC molecule. Now once this is tepsin, let me write tepsin. Okay, when it is bind with this tepsin proteins, so they can break down or they can remove out this ERP57 proteins. Okay, they can break down this chaperone, and they can remove this chaperone as well as they can remove this ERP57 protein to allow the antigen to bind that specific MHC1 molecule. So antigen are then, or we can say the amino acid are then easily bind to this MHC1 molecule. So after some time we get a free and complete MHC1 molecule through which the antigen or through which the amino acid are bind. Let me draw here. So here the antigen are bind, or we can say the amino acids are bind. Okay, let me draw the amino acid here in the black dots. So once the amino acid are bind here, so then it is shifted, or it is then removed out from the endoplasmic reticulum. So it is removed out from the endoplasmic reticulum, and then it is passes from the Golgi body. Now once this complete MHC1 molecule having the amino acid or having the antigen are passes from this Golgi bodies, so then they make endosomes. Antigen. Okay, so once this is the endosomes. Now after that, these endosomes are then fused with the cell membrane, and then this MHC1 molecule are expressed on the surface of eukaryotic nucleated cell, or we can say it is then expressed to the surface of the nucleated cell. So after some time, this MHC1 molecule, once it is expressed, so now, so what actually happened? This MHC1 molecule are expressed. Alpha 1, alpha 2, alpha 3 and beta 2 globulin. Now once this MHC is expressed to the cell membrane of the nucleated cell having the amino acid or having the antigen, so after that what actually happened? Now this cell is infected. This cell is what? This whole cell is now infected. Let me write here, this is infected cell. Infected cell. When the cell is infected by this bacteria, after that what actually happen? They will bring or they release some kinds of cytokines to call upon the cytotoxic T cell, or we can say CD8 cell. Let me draw here CD8 cell. Okay, so this is what? This is the CD8. CD8, or we can say cytotoxic T cell or cytotoxic T lymphocytes. We can also say that like that. Now, these cells have also their own receptor, which we call that CD8 receptor and T cell receptor, cytotoxic T cell receptor. Let me draw each of the receptor here. It is made up of alpha and beta subunit, and this is also made up of TCR is also made up of alpha and beta subunit. Now once this CD8 cytotoxic T lymphocyte or cytotoxic T cell recognize this antigen, and this is means infected cell, so after some time these cells will produce a kinds of granzyme and perforin to this cell and cause the destruction of this cell. So finally these cells are destructed, or we can say these cells are killed by the cytotoxic T cell to reduce the chances, to reduce the infection chances. Okay, why? Because once this cells is infected, so again another cell, there is a chance of another cell to be infected. That's why the cell is stopped, or the infection chances will be stopped. Okay, so after that the process will be under the control. So let's say this is what? This is the CD8 cell. So this is not a complete structure of the CD8 or cytotoxic T lymphocyte. So inshallah I will draw the exact structure of cytotoxic T lymphocyte in some other video lecture. Okay, so that's how the synthesis of MHC1 molecule. That's how the MHC1 molecule are synthesized and processed. Okay, so let me once again tell you that we have a pathogenic bacteria. It is, internalized. It is not engulfed. Okay, remember, it is not engulfed, it is just internalized. Then once it is internalized, so the cell is infected. Once the cell is infected, after that this bacteria will produce the endogenous proteins. Now this endogenous protein are passes from the proteasome. Proteasome break down it into small fragments again, and we get a 15 amino acid. So these are the 15 amino acid. After that, these 15 amino acid are passes from the endoplasmic reticulum, rough endoplasmic reticulum. Now after some time, these amino acid are further hydrolyzed, and we get 8 to 10 amino acid. Now after that, these ribosomes are activated and they make the protein or the product of MHC1 molecule. Now after that we get the incomplete MHC1 molecule. This is the incomplete MHC1 molecule having the weight of 43 kilodalton. And there is another beta 2 subunit are also synthesized inside the endoplasmic reticulum, and it is bind to this MHC1 molecule to make a complete MHC1 molecule. Moreover, this incomplete MHC1 molecule have a chaperone-like structure which we call it chelanexin. Now chelanexin will just present here, and they will just bring the MHC1 molecule to that endoplasmic reticulum, or they are just shifting the MHC1 molecule inside the endoplasmic reticulum. After some time, there is another protein which we call that ERP57. Now ERP57 will just bind, or they will lock this MHC, the binding side of the MHC1 molecule, the antigen binding side of the MHC1 molecule. After some time, this complete MHC1 molecule are shifted to the tapacine protein. Now this red one is the tapacine protein. After some time, again this MHC, the chaperone and this F57 will release out, and we get a free MHC1 molecule having the antigen binding site. So amino acid or the antigen are bind to this binding site, and after that they are eliminated or released out from the endoplasmic reticulum. From the endoplasmic reticulum they are passes from the Golgi body, and from the Golgi body they are converted into the endosomes. Now inside the endosomes we have the MHC1 molecule having the antigen. After that they are passes from the cell membrane, and from the cell membrane then this nucleated cell membrane which is infected, they expresses this MHC1 molecule, because we already talked that MHC1 molecule are always present in all nucleated cell. Now after that, once they are representing this this antigen, so the cells are activated, which we call that cytotoxic T cell. They release a kind of cytokine due to which the cytotoxic T cells are activated. Now this cytotoxic T cell have their own receptor which we call that TCR, T cell receptor, and we have CD8R or cytotoxic T lymphocyte or CD8 receptor. Now both receptor can recognize this MHC1 molecule. Once they recognize this MHC1 molecule, then they produce granzyme and perfurins. So they produce what? They produce granzyme and perfurins. Now this granzyme and perfurin will cause the programmed dead cell or apoptosis, or we can say this cells finally killed. Okay, so that's how the MHC1 molecule are synthesized. I hope you get the idea about the MHC1 molecule, the synthesis of MHC1 molecule. If still you have any kind of question in your mind, you can write it into a comment. Inshallah, I will give you answer as soon as possible. So if you like this video, make sure to hit the like button, share this video, and stay tuned, because in the next video lecture, we are going to talk about the synthesis of MHC2 molecule. Thank you so much for watching.